Hello everyone, my name is Maxwell Mkunga. My specialty is meat science and muscle biology, especially with small ruminants. And so today we are going to talk about goat production. So that's my outline. We are going to talk about the history of goats in the US, how we manage these goats, and then I'm going to talk about LiDAR scan experiment. So goats were among the first domesticated animals, and this was due to getting products like milk, meat, mohair. So US goat population currently is 2.5 million, with 80% actually being the meat goats. Texas predominates in the production of goats. So this is a graph showing the top goat meat producing countries. You see China, followed by India, Pakistan, Nigeria, Ethiopia, with more consumption reported in Asia, Africa, and the Caribbean. Some of the goat terminology that we have include genus, that's Capra, species, Carcass. Commonly the terminology is caprine. The key terms for a male goat, that's a buck, female goat, doe, and then a young goat, kid, and a castrated male, a wether. Also, the sheep shares this name. So for the meat, you will hear chevron, which is an old goat meat. And then for those young ones, we call them cabrito. Goats are reported to be seasonally polyestrous, and what that means, poly means many, estrus means on heat. So they come on heat multiple times in a certain season. So normally we do breeding here around fall, and then we wait for kidding in the spring. Meat type goats generally are weaned to feedlot at 1 to 2 months of age, around 40 to 60 pounds, and we normally wait for the market weight around 100 to 125 pounds. So these are just example of breeds that we have at the TU farm. That's a boar goat and a kiko. So Spanish goats, they first arrived in North America with the Spanish explorers. So these were the first ones to arrive. They are very hardy and can thrive in difficult environments. I use them in my master's research, and they are very good mothers and also hardy when it comes to parasites. They are very resistant to these parasites. The Kiko goats were first imported in 1992 from New Zealand. I've also worked with them. This picture is in Mississippi, where normally producers donate some of the bucks to try and see their performance for improving breeding. The boar goats were first imported in 1993 from South Africa. They are very good for weight gain and meat quality. So normally they have a high dressing percentage, which we are going to talk about later. After the boar goats, we have the Savannah goats that were first imported again from South Africa in 1994. They are usually white. You can see they are characterized by white coat because this coat allele is dominant over the others. Goats are exposed to various infectious diseases as they live in a variety of microbe-rich environment, which poses a significant threat to them. Some of the pathogenic and parasitic problems we have in the goat production include mastitis, although this one is more on the dairy side. We have foot rot, which is a bacteria disease causing lameness, and so it's very contagious. We have gastrointestinal nematodes, and specifically Haemonchus contortus, also known as the barber pole, which negatively impacts the goat health. Some of the control measures that we have include chemical-based drugs like anthelmintics, antibiotics. We see some research also being done on probiotics and prebiotics. Some use plant-based anthelmintics, example Sericea lespedeza. They have been seen to be able to improve the immunity of the goats, and that way you don't have to rely on the chemical-based drugs. I also did my master's research using medicinal mushroom, Ganoderma lucidium, also known as reishi mushroom or lindsay mushroom. So it has those bioactive properties that are able to improve the immunity of the goats. We also have vaccines, and the type of breed also determines the resistance when it comes to these parasites. Like we mentioned, Spanish goats are very resistant to these parasites. And so how do we monitor the health of these goats? We normally use Farmacia card, which was developed actually in South Africa to identify anemic goats. As you can see from the chart, normally goes to a range of 1 to 5. Normally 4 and 5 would identify the goat as anemic. So we also check the body condition score by checking the fat and the muscle distribution at the spine and also at the back of the goat, which tells us about the health of the goat. We also check on soiling at the tail, check if there is any swelling at the jaw. We check the nose for discharge, among many others. And then we do fecal egg count, which is like a confirmatory test, which is a flotation test that separates the parasite eggs from the debris based on density. And so when you use the microscope, you're able to count the eggs of these parasites. We have talked about producing these animals and also monitoring their health. Let's now shift gears and talk about how do we monitor growth and also efficiency. So some of the changes that we look for include body weight, which is one of the first measurements you see, although it's not always the best way to assess growth because you might have shrunken weight and also live weight. Shrunken weight is when we fast these animals around 12 hours to 24 hours, which is normally the accurate weight. For meat animals, the most important measurement is carcass weight, which is measured after harvesting the animals, and then we remove the offals by removing internal organs, feet, and head. We also have dressing percentage, which is calculated by dividing the carcass weight by shrunken live weight. It depends with the breed. So goats are reported to have a dressing percentage of 50%, but depends with the breed. Some breeds, like I mentioned, bull goats, have around 55% compared to Kiko, which might have around 48%. We also have carcass measurements. We have carcass length. Longer carcasses are associated with more lean tissue. We have longissimus muscle area. Longissimus is Latin meaning longest. It's one of the largest muscle in livestock. It's used to measure excess muscularity and muscle growth. We also have subcutaneous fat thickness used to assess fat levels in an animal. And so let's talk about LiDAR experiment where I did comparison between manual scan measurement and LiDAR scan on beef carcasses. And so what's a LiDAR? It's a light detection and ranging sensor technology that uses laser light to measure distance to objects. Apple introduced in their products in 2020, you can see in iPhones and also iPad. So the light travels from the sensor to the object and then bounces back to that sensor. And when you have a 3D application like Polycam, you're able to get a 3D image. Some of the applications that we see with LiDAR include self-driving cars. We see it being used for drone-based fertilizer spraying. They're able to map the topography of the land, and in that way they're able to calculate the height of crops. So farmers are able to apply fertilizer efficiently. In this third paper, they were measuring athletes' lower limb, that was the shank and the thigh, and they reported a relative coefficient of determination of 83% and 90%, showing a strong correlation between LiDAR and manual method. So you can see one of the images has distortion, and they reported that lighting affects LiDAR, and so I had to factor that before my experiment. In this other paper, they were measuring live animal, trying to predict the sellable cut, and they reported a relative coefficient of 72% showing accuracy of LiDAR. Whale biologists spend most of their time on boat just to get a glimpse of the whale. These kind of big animals, you always see them from a distance. It's in 2021 whereby a big female whale was found near the beach of Alaska, and the University of Alaska Southeast, they were able to use a drone-based LiDAR to take the 3D image of the whale. And when they posted in their Google site, it accrued a lot of interest from all over the world, from researchers, scientists, showing the importance of this kind of technology. And so I was so interested and wondered how I could be able to use in mid-science. As I mentioned before, taking the carcass measurement is very crucial for the processor in planning ahead and also for efficiency. And so our objective was to compare the manual method and the non-contact LiDAR scan measurement on the beef carcasses. So this is the user interface of the LiDAR using the polycam, and you can do some of the linear measurements. Also, there is part for the area, and you can measure area. We also have AR, which is augmented reality, and you can be able to put these carcass on real time. We hypothesized that there will be no significant difference between manual measurement and LiDAR scan measurement on the beef carcasses. So we used 5 bovine carcasses which were split into right and left side, so we had a sample size of 10. For the manual measurements, we used a tape measure for the carcass length. We used a USDA grid ruler to assess the thickness of the back fat, and a USDA dot plastic grid to measure the ribeye area. For the LiDAR scan, we use a polycam for the 3D image. We use iPad 11 and iPhone 14 Pro Max. The scans were performed by one examiner moving at a constant speed on a circular path around the carcass for approximately 2 minutes, making sure that the iPhone and iPad were kept at as much as stable, keeping a distance of about 50 to 100 centimeter. Then I used paired t-tests using SAS to analyze the difference between the two methods. Some of the measurements that we did include the inner length of the carcass, the outer length of the carcass, the ribeye area, which we normally split the carcass at the 12th and the 13th rib. That's where you get the ribeye. It's normally one muscle, longissimus muscle, which I mentioned earlier, and that's where you get most of your steaks like ribeye and New York strip. We also measured the back fat thickness, as you can see. And so in our results, for the linear measurements, that was the whole carcass length and the inside length of the carcass, there was no significant difference between the two methods, which is what we hypothesized. For the back fat thickness, there was also no significant difference between manual measurement and LiDAR, which is what we hypothesized. It's only the ribeye area, as you can see the p-value, which was significant between the two methods. We started with the iPhone. As you can see from the second picture on the polycam, when we were trying to draw the area using our finger, we caught a thick line which was not precise, and so we hypothesized that using an iPad, using the Apple Pencil, we could be more precise. But again, we tried and still we were getting that thick line, and so that's one of the limitations on the polycam. And so we are trying to explore other specialized web-based 3D analysis tools that we can import the LiDAR OBJ file and maybe try to get the area. Also, for future direction, we want to see the applicability of LiDAR on different species, especially the small ones like goats, sheep, pigs, among many others. Some of the advantages of LiDAR, like I mentioned, is non-contact. You don't have to touch the carcass, and in a food safety perspective, that's good. It's also faster and reproducible compared to the manual measurement. You also get a digital file, OBJ, which is good for traceability purposes. We also see a future where USDA can work remotely instead of being on site. You can just send them a 3D image for them to evaluate the carcass. We are also planning to buy virtual reality headsets and try to use this technology in training students in 3D learning in meat science and also anatomy class. So in my conclusion, LiDAR shows a great potential in modernizing carcass evaluation, and it's time not only in meat science, other fields to also embrace these digital tools for precision, efficiency, and better traceability. Thank you.